Hello everyone. Now I am starting my class. Okay, so we are continuing our class from where we stopped in the last class. So we discussed about sources of drugs, how to find new drugs, and what is the need for developing new drugs and all those things. In this class, we are going to discuss how to develop the new drugs. Okay, it's going to be very interesting because what is the importance of this as far as academic point of view as an MBBS student? MBBS students need to know how the drugs are developed, what are the different phases of drug development. It's important because these MBBS students can be future doctors, and they are supposed to know how the new drugs are developed. And whenever a medical representatives come to you for introducing a new drug, you should be able to ask for what are the different phases have been already covered and what is the status of approval of this drug in the market. Because someone, if you don't know these steps, someone can easily bluff you that this drug is already approved and this is what has been already done, and they, if even if they have not done, they can bluff you. So it's important for you to ask the data that has been developed or approved for that particular molecule over a period of time. If you know, for developing one drug, it may take more than 10 years. So any Tom, Dick, and Harry should not be able to come and bluff you that this drug has been developed and it is useful for so many patients. But it is important for you to know which is the drug properly classically developed and which is the drug which is not yet been developed. Because suppose if you use a drug which is not approved, then if patient dies of any adverse drug reactions, then nobody will be there to protect you. You will have to substantiate yourself. So, and sometimes what happens, MNCs, they develop the drug in different countries and they come to India and say that this drug is available in India, can you use it? But you should know that every drug should get approval in the particular country according to the regulatory processes developed in that particular country to ensure that all the safety features are taken care before allowing it to get marketed in that particular country. So every MBBS doctor should know the processes of development in that particular country. Okay, how a new drug is developed in that particular country. They should be aware of that. Then only they will know that, okay, whether the drug is properly approved or not. In our country, if you know, Drug Controller General of India, he is the supreme authority for approving the drugs in India. There is something called as organization, that is CDSEO, okay, Central Drug Controllers Control and Standard of Drugs Organization. Okay, so they maintain the list of drugs that are approved in India. They will keep on publishing particular drugs that are approved in India. Maybe if you go to their website, you will see that there are list of drugs which have been banned in India also. Okay, in the CDSCO website, you will be able to find such list. Okay, recently you might have seen some news clipping saying that more than 20 drugs, FDCs, have been banned in India. So should you use these banned drugs or not? You should not use. If you use, suppose if any consequences happens to the patient, then you will have to, save yourself. You will have to fight for yourself. Nobody will be able to help you. Okay, so with this background, I would like to tell you that you should know the, at least not in detail, if not in detail, if you know little bit about how a new drug is developed, then at least my purpose is served. In addition to it, what is the use of knowing this process as far as examination point of view? In the final examination, you will have some spotters coming from this lecture. What are the different phases of clinical trials? What are the different ways in which a new drug is developed? Or in the final examination theory also, you may get some spotters, you may get, and also some charts will be available, you may have to answer. And also in one of the short notes, it may take some space there, they may ask you to highlight what are the processes that are followed in phase 2 trials, phase 3 trials, or phase 4 trials, pharmacovigilance. So they may ask little bit, maybe short note 5 marks question or 3 marks question with regard to this lecture. Okay, so let us try to discuss how a new drug is developed. Okay, what are the stages of new drug development? Okay, so if you know here, you can see that there are four phases or four stages of new drug development. First stage is in vitro, in vivo testing of new chemicals, new chemical entity. So or investigational new drug, uh, new drug. Okay, investigational new drug. Okay, IND, or new chemical entity. Okay, so NCE, we call it as NCE, new chemical entity, or investigational new drug. Okay, so once a new drug is identified, new molecule is identified, so they will do some basic research. Okay, you can see the here basic research, like what is the chemical properties of it, what are the, like whether it is soluble in water, whether it is soluble in alcohol, or what is the chemical structure of it, what are the, how it is different from already existing molecules, whether this can cross the, like theoretically, whether this can cross the blood brain barrier, or whether it can be easily observed or not, like that. Some things, some information related to this through the basic research can be obtained. And then early discovery activities will be followed by this, which involves like testing in the in silico models, and also evaluation of pharmacokinetics, pharmacodynamics in silico or isolated cells. So like that, so that they may do that, and then they will derive some pharmacokinetic and pharmacodynamic parameters that can be probable. Okay, what are the probable pharmacokinetic, pharmacodynamic parameters that can be expected? Then after that, this is about proof of concept. Okay, so they will do the, like in which condition it can be useful, or which is the cell it is expected to affect, what kind of, whether it is affecting the pancreatic cells or gastric cells or CNS, in CNS which place, so or any other cardiovascular system, whether it is blood vessels or it's a heart. So like this, like which is the targeted molecule, targeted organ, targeted tissue, targeted physiological process. So they will try to identify that, and then they will also try to identify which route is the most suitable, whether it is orally absorbed, or whether it can be given intravenously. Is it irritating the tissue or not? So like that, yeah. So like that, they will try to evaluate, and how easy it is to reach the target site. Okay, so like that, they will try to evaluate, and then they will formulate Okay, so formulation will be done accordingly. Okay, so if you want to give by orally, then they will give the active pharmaceutical ingredients, API we call it as. They will synthesize in large quantities and ensure that it is stable in the particular formulation. They will have some excipients they will add to make it a good salt so that it is stable in that particular form. Okay, so then they will start doing the preclinical studies. Preclinical studies means it is the animal studies. Okay, so animal studies means, so you either at least two rodents will be considered immediately, initially, and then one rat, one mice, or any other, like guinea pig or rabbit. So like that, they may consider some important animals who are which are regularly used for the purpose of research, and also they will try to give the drug in a particular route according to what has been expected. So they will, what they are going to test, they are going to test the pharmacokinetics, that is the absorption, distribution, then metabolism and excretion, deposition in which tissue it is going to get. So all these things, all basic information they are going to get. So once that is done, they will also develop the models, various models. Like suppose if it is CNS disease is the target, then CNS related related tissues will be considered for related models will be CNS related models will be considered for evaluation. So CNS models means maybe in a rotor rod or a pole climbing operators or any maze, so like this. And for every for every disease, for every target organ evaluations, there are different kinds of models are available. So they will try to evaluate using that, and also they will do the isolated tissue experiments, isolated cells, cell lines experiments, to evaluate varieties of activities like tissue toxicity or adverse drug reactions, capability to develop the adverse drug reactions, and then teratogenicity, carcinogenicity, and then so like the tissue sensitivity, tissue toxicities, so tolerance or cross tolerance. So like this, varieties of evaluations are done for that particular molecule. And once we get convincing evidence in preclinical stage, most importantly, it should be safer. Okay, it should be safer. It should not be carcinogenic. It should not be genotoxic. So it should be of good quality. It should have some good quality. So after that, the more molecule will move to the animal testing. You can see here there is something called as IND application. Okay, IND application means investigational new drug application. Once it successfully passed through the basic research, early discovery, and preclinical testing, then there will be a application will be made to the regulatory agency saying that we have invented this, and they will get a patency for this, and they will say that we have we own this molecule, we would like to develop this. Please give us further permission to go ahead with the evaluation of the molecule for further development. Okay, then you can see that human development, human testing will start. So in this you can see here there are four phases of development here. You can see phase 1, phase 2, and phase 3, and then phase 4. Okay, so you can see here there are three phases are in green, and the another phase is in light blue, navy blue. So why is that? So you can see here phase 1, phase 2, phase 3. These trials are mandatory for getting the marketing approval. Okay, marketing approval. This clinical development is mandatory for getting the clinical approval. You can see for the marketing approval. Okay, you can see either phase 1. Generally, healthy volunteers. Okay, healthy volunteers are considered for evaluation. Okay, so it can start from 100 to 10 volunteers, maybe 1 2. You can start with 1 2 volunteers, and then you can increase to, you know, so 10, 15, 20, then 40, 100. Okay, up to 100 patients. Okay, up to 100 volunteers, healthy volunteers, we can consider in the phase 1 trials. What are we going to do in this phase 1 trials? In the phase 1 trials, we are going to evaluate the pharmacokinetics and safety. Okay, pharmacokinetics and safety. What is pharmacokinetics? Pharmacokinetics involves absorption, distribution, metabolism, and excretion in humans. Okay, if you see here in the preclinical stage, it is done in animals. Okay, so considering that, they will decide the dose. Okay, they will decide the dose, that is the 1/10th or 1/100th the dose that is being tested, a safer dose that is tested in preclinical, that will be considered for the purpose of analysis. And also, of course, based on the amount of expected response. Okay, what is the dose range that is suitable for getting that particular response, so accordingly also it will be considered. But in phase 1, definitely it is, they will start with 100th dose, and if it is okay, then they will go to 1/10th dose, and then they will see whether they can reach the maximum, and they will also do the dose escalation studies. Okay, so single dosing study, after that multiple dosing studies, like this they will continue the evaluation of the particular drug for the purpose of taking to the next level. So once the drug is found to be safer in healthy volunteers, then it is important for us to do the testing in the targeted patients. Okay, targeted patients means which disease, whether it is in the myocardial infarction patients, whether it is in the allergic rhinitis patients, whether it is in the Alzheimer's disease, like that, like, or in a particular cancer. Okay, so they will try to evaluate in a small number. Okay, maybe 100 to 200 to 300 patients. Okay, so patients I'm saying. Okay, earlier it is in the phase 1 it is healthy volunteers. Now in the phase 2 we are going to evaluate in patients, particular patients whom we would like to target. Okay, so targeted patients will be selected based on the inclusion exclusion criteria, and most importantly, most safer criteria will be considered for this. Okay, because extreme patients may not be considered in this phase, but try to mild to moderate or severe patients with no seriousness, they will be considered generally. And then further, like, what are we going to evaluate here? Here again safety will be considered, and also dose range calculations will be done, and again safety will be evaluated, and pharmacokinetics may be little bit, since it is patient studies, then they may do population pharmacokinetic studies, that is absorption, distribution, metabolism, and excretion, little bit they will do. But main objective is to see whether the particular disease what we are trying, in that case, in that disease, whether there is any efficacy or not, whether the particular drug is better than placebo or better than the existing drug. So like that they will try to understand, and once that is done, then they will move on to the, once we find in less than 300 or 100 to 300 patients, if you find some safer use and also good response is obtained, it is okay. If we are okay to go to the next level, then we are going to, generally in phase 2 trials we will do in one ethnic group. Okay, so it may be in the same country. In India, maybe 4 to 5 centers can be there, and north, south, so east, west, and also we can have one center here and there, and then we can collect the data and accumulate and make a report of it, maybe 100 200 patients, or even in the other countries also they may do. But in when we get sufficient evidence in phase 3 2 trials, then the drug will move to the next phase, that is the phase 3 trial. Okay, in phase 3 trial, what are we going to evaluate? In phase 3 trial, we will continue to evaluate what has been evaluated and confirmed in phase 2, and the same thing will be done in phase 3. Then why we need to repeat this trial? Because it will be done in different ethnic groups. Okay, different ethnic groups, different countries also. Okay, so it can be like, so throughout the world also they may initiate the trials, and also different conditions. There may be 10-15 trials they may plan, one after the another, with the different clinical conditions. Maybe one with mild patients, another with moderate patients, another with addition of one drug, another with without the drug, without the comparator, with standard control. So like this varieties of trials, they may try to ensure that in different countries, in different ethnic groups, in different clinical situations, what are the benefits we can get for this patient by using this particular new drug. How is it doing in comparison to the existing treatment? So all these things will be evaluated, and also to find the whether it is better than the existing drug and safer than the existing drug, and whether this can be given to the patients. No, so like that. So whether there is any benefits, real benefits are there or not. So, and how many patients will be evaluated in this? In phase 3 trials, usually it will be in thousands, maybe 1,000, 2,000, 5,000. It can go up to 10,000 patients also. Okay, so this is mainly to satisfy the regulatory requirements, so that they can get the new drug application. They can put a new drug application, so that after the review and approval, the drug can enter into the marketing phase. Okay, so once the drug is found to be safer, effective, then the drug will move to the marketing phase. So marketing phase, after it enters, like the successfully completes phase 3 trials, it means that it can go to the next level, that is marketing phase. So as a MBBS student, you should know that you should ask the medical representative, whoever come to market that particular drug, you should ask for the all these data, phase 1, phase 2, phase 3 trial data, so that you can ensure that whatever the new drug you are going to try, it is already passed through the regulatory hurdles. Okay, after that, like what you are supposed to do once the drug is in the market, that the thousands, lakhs, or millions of doses will be used in the market. Okay, at that time what can happen? So lot of adverse drug reactions can prop up, and you can see that there are so many drugs which are removed from the market within 1 to 2 years after the marketing. Okay, because so many adverse drug reactions in the controlled conditions, especially in the phase 1, 2, 3 stages, they were not identified, or if they are identified, not properly projected, not informed to the regulatory agencies. If it happens, like like terfenadine, it came into the market, it got removed immediately because of cardiac adverse drug reactions. Gatiflaxacin, it came into the market, it was again removed because of the cardiac adverse drug reactions, hyperglycemia, like that. So then, so similarly, lot of drugs were thalidomide, it came into the market, it was removed again. So like that, like lot of drugs crosses different stages, but they get removed because of the safety reasons. That's why the phase 4 stage takes very important. That is, the phase 4 means post marketing surveillance. Surveillance means we continuously get the hotspots, or any adverse drug reactions, or serious adverse drug reactions, or mild adverse drug reactions. All these are logged into the regulatory software, and they will have a watch on all these marketing experiences. And regulators mandate the marketing authorization holders to regularly update the safety status of it. Every 6 months there will be post-marketing surveillance report, that is PSUR we call it as, update report they have to give for the first 2 years in India every 6 months. After that, to the next 2 more years, they have to give once a year. It means that totally 6 reports in 4 years they are supposed to give. Okay, so this is what is the new drug development stages. So I believe that I have explained all the different stages. So the fourth stage we can call it as even pharmacovigilance, the active pharmacovigilance we call it as. After the after the drug becomes the world, then the regulators will become lesser stricter. Okay, then there will be a continuous passive surveillance will keep on happening for all the drugs once they are in the market. Okay, now I'll try to quickly recap what I said. So once the drug is discovered and developed, it will get into preclinical stages, and then it gets into clinical stage, and then it will be reviewed and it will be allowed for marketing. Now, I think I have explained to you that like different phases will have different goals, and they will try to identify the targets and optimize the targets, remove the drugs which are not properly meeting the targets, and then generate the molecules which are based on these failures. They can again rework on the molecule, generate the good molecules, and again they will keep on developing, and better molecules will enter into the preclinical stage. In the preclinical stage, again, as I said, isolated cells or full animals, like in vivo testing or in vitro testing can happen, and then molecular docking mechanisms, evaluation, all those things will happen. And again, they will do the toxicological testing and consistency assays, and then they will do the filing of investigational new drug status they will get, and then they will allow it for going for the development of drug in the humans. Okay, so this is the, I think, in the preclinical stage, what are the activities they will do. You can see here that's the primary pharmacodynamic screening will be done, then pharmacokinetic studies will be done, toxicological studies will be done. What are the toxicological studies they can do? That is acute toxicity testing and assessment of safety will be done. Subacute and chronic toxicity testing will be done. Local irritant effect studies will be done. Special toxicological studies like reproduction related toxicity studies, teratogenicity studies, mutagenicity studies, carcinogenicity studies will be done. So this is what I have already mentioned. Now, if you see the preclinical clinical testing, once we have a successful preclinical trial, then there will be IND submission will be done. Then phase 1, phase 2, phase 3, phase 4 will be done. Just to recap what I just said, in phase 1, generally healthy volunteers will be done. It can start from 20 to 100 volunteers can be there in basically PK/PD. That's the pharmacokinetics is the most important evaluation they will be doing. In phase 2 trials, average 2 years is the required time, and in this safety and efficacy both this especially this testing, phase 2 testing is done in patients, and safety of the molecule and the efficacy effectiveness in the particular disease will be done. And then once successfully phase 2 is done, then phase 3 will be started. In this phase 3, the number of volunteers can be few hundreds to few thousands volunteers can be there, and it can take on an average 1 to 4 years in developing this. Again, whatever we considered in phase 2, the same extension trials can be done in phase 3 with multiple ethnic groups in the multi-country trials. Once we have some convincing evidence, then new drug applications will be filed, and of the approvals, like in India DCGI approval will be obtained, or in US FDA approval, or in UK MHRA approval will be done, or European Medicine Agency, EMA we call, so EMA approval will be obtained. So as I said, all the requirements you have to meet as per the clinical development, phase 1, phase 2, phase 3, safety, efficacy, pharmacokinetic, dynamic. All these things will be evaluated, and finally we can move ahead with the development, with the approval. Once the drug is approved, it will be again, post marketing surveillance will start, and safety marketing approvals will be like, that is the safety updates will be provided to the regulatory agency, as I mentioned. Okay, so there will be differences between the small molecules versus biologicals development, which I'm not going to discuss now because it is out of your syllabus. And drugs for pediatric use also separately drug need to be developed. Generally this group is like a pregnancy, and pediatric groups are not included in the regular development. That's why special development need to be done in these groups. And again, kidney failure patients, liver failure patients, again special kind of attention is required. So again, separate trials need to be conducted to ensure that in these groups also drug is effective. Okay, so what are the important guidelines what we have in India? You can see here that Indian GCP 2001 it was developed. Indian GCP means Good Clinical Practice Guidelines. See, this guideline is very important as far as the development of the clinical development of the drug. Okay, so all the investigators throughout the country, they are supposed to follow this guideline. Okay, GCP guideline. I'm going to tell you in the next class about the what are the GCP, some 13 commandments or the important requirements of GCP. So that I'm going to discuss in the next class. In this class, I'll just try to introduce about the Indian GCP, Drugs and Cosmetic Act 1940, Drugs and Cosmetic Act 1945, and the Schedule Y. It is the part of Indian guidelines, but now it has been changed. Okay, so Schedule Y and all the all these guidelines has been integrated into new clinical trial. Okay, new clinical trial guideline that was released in 2019. Okay, in India. So that's the Drugs and Clinical Trial Rule 2019. So which is available now for this purpose. And again, ICMR ethical guidelines for biomedical research on human subjects. This is the another important guideline which is available in India. So all the research and development and clinical research or clinical any kind of research that happens, it should follow this ICMR ethical guideline. So without that, we are not supposed to conduct any human research. Okay, so this is about the what are the and some more guidelines, that is international guidelines, or Helsinki Declaration is there. It was developed after the thalidomide disaster in 1964. So that was World Medical Association. Okay, World WMA. When World Medical Association was developed in 1964 after the thalidomide disaster, and they will every 4 years they will keep on meeting, and if any regulatory lacunae are there, they will keep on regularizing these lacunae, and then they will add some guidelines. So it is called as Helsinki Declaration. Okay, in 1964, first time World Medical Association met in Helsinki. That's why it is called as Helsinki Declaration, and it's a very important guideline requirement. Okay, so this requirement need to be mentioned in every clinical trial protocol or clinical research protocol, that like we are going to follow this Helsinki Declaration. Without that, we are not supposed to conduct any research. So okay, so that's why I have tried giving you some crisp information about what are the important regulations that are available in India, and you should be able to, you should be able to adhere to all these. If you are suppose, like, see, every one of the one of the requirement for an Indian medical graduate, IMG, he should be a continuous learner, and he should be also a researcher, keen observer also. Okay, so ensure that he understand the latest requirements of that particular country guidelines, and ensure that he is part of that particular learning process. Okay, so since he is going to be the decision maker in healthcare, as far as healthcare concerned, and he will be a team leader. Okay, he will be a team leader as far as the health management is concerned, and he should be able to take independent decisions in managing the patient's health. He should be aware of all these guidelines, and ensure that like the person, he or she, whoever, okay, Indian medical graduate, should be able to understand all these guidelines and follow. Suppose if a medical representative come to you and say that I would like to introduce this new wonderful drug, but if we start talking in such language, then you should be able to ask for the literature. What are the publications you have? What are the approvals you have? What are the, whether it is approved in India or not? Is it banned in any other country? So like this, you should be able to ask these questions, and then try to find what is the latest available information on that particular product, and try to go to be independently also. You should be able to collect the information from the right source, like I would say PubMed. You can get into PubMed, or varieties of scientific journals are available. So you should be able to get into the websites, and now it is in the tip of your hand. You have all the information. You should be able to try and find out what is the current latest information available, so that like nobody can bluff you. Like because they may like generally business, like business interests, sometimes they go out of the safety standards. Okay, so if such things happens, then you being the decision maker, you should not like put your patient's health in the risky side. Okay, you should try to protect your patient's health. So if you want to do that, then definitely you should be aware of what is the latest information available about that particular drug. What is the current status? I think, yes, today I received a few safety information from DCGI and the Pharmacovigilance Committee, Pharmaco PVPI program. They mentioned that like a few drugs like ceftriaxone, it can cause some cardiac adverse drug reactions. So like this, like there will be latest updated information. It will keep on coming, and it will be updated in the websites of WHO, Drugs Controller of General India, the CDSU website, or ICMR website, or it will also be shared in the networks like LinkedIn or any other areas. So you should be vigilant. Like whenever you get some information, you should try to find out the facts, whether the information what you received is right or wrong. If any issues are there in the information that we received. Recently I saw one myth circulating in the group that like some nursing council has issued. So it was not issued by the nursing council, it has been cleared. So like this, like some misconceptions, misinformation will be spreading in and around. We should authenticate, like we should check the authenticity of the information what you're receiving. So to know that, to do that, you should be knowledgeable enough to what to check, how to check, where to check. So these things. Okay, so I will stop now. So we have only 1 minute. In the next class I will continue with the good clinical practice guidelines, and also there will be a quiz in the end. Okay, so that's what I'm going to do. So I will stop here. Okay, so all the best. Anyone else who is there here? We have Navya and Shraddha. So hi Navya. Hello Navya.
Yes, sir.
Okay, you are able to get what we are
Yes,
discussing,
sir.
what I'm discussing. Do you think that this is useful?
Yes, sir, it is useful.
Okay, Shraddha is there, or anyone else is there? Okay, so I will stop here. Okay. So in the next class, I will again I will let you know when I'm going to take. Inform everyone to join the class. Okay. So and I will send the link also. You people can, those who could not make it, they can visit the link and attend the class. Okay. So all the best.